Yeah, so maybe we'll go ahead and get started. Yeah, so it's great to see everyone here today. I'm Justin Colsino. I'm faculty in environmental health sciences at the School of Public Health. I'm a member of our single cell spatial analysis program leadership team. And so just a little bit of housekeeping before we get started. Many of you have probably already signed up to join our program as members, but if you haven't and you're interested, we can add a link in the chat to our website where you can learn more about the program, see what it's like, and then follow for a link to a short membership application. And so it's my pleasure today to have the opportunity to introduce our speaker, Dr. Johnny Sexton. So Dr. Sexton did his PhD in chemistry at UCSD and his postdoc at Stanford in structural biology. He joined us here at Michigan as faculty back in 2018. And so he has faculty appointments both in internal medicine and in the medical school and in medicinal chemistry at the College of Pharmacy. And Dr. Sexton also leads the Center for Drug Repurposing. And as you'll see today, Johnny has a very impressive research program in the area of drug discovery, development, and repurposing. And of particular relevance to this audience are his lab's effort in developing high content imaging-based approaches for drug screening. As you'll see, Johnny and his team have developed imaging-based assays that allow them to take like thousands of morphological measurements at a single cell level to really understand how drugs or stressors start to perturb cellular biology. And then you also see how his group has developed some pretty cool advanced artificial intelligence platforms to really make sense of all these complex measures. And then as you can see from the title of his talk, his lab is applying these exciting methods to a broad range of different disease outcomes. And so we're psyched to have Johnny here to present his work. So if you have questions during the talk, feel free to add them into the Q&A. And hopefully we'll have some time to discuss after Johnny's presentation. With that, my pleasure to turn it over to Johnny.
Thanks a lot, Justin. Appreciate the introduction. And please, interrupt me if you notice some questions in the chat or something like that. Interrupt me right in the beginning. I hate railroading through talks, just from front to back. So please feel free to interrupt. Okay, thanks. So I'm going to tell you about our technology platform, which collectively is sort of all the tools of modern molecular and cell biology, fluorescent probes, high throughput screening, laboratory automation to do drug screening, drug discovery, drug repurposing, coupled with computational technologies. And so I put my close collaborator, Matt O'Meara here. This is really a joint effort between our two labs. Matt's in DCMB. He's also very interested in drug discovery and artificial intelligence, machine learning methods. And so he also is a very recent faculty here as well. And so we had these aligned interests and have really done, developed tools that sort of go hand in hand where, we're doing the high throughput automated imaging, phenotypic characterization, really robust assay development. So we get highly robust endpoints, but then layering on top of that, the machine learning models like Justin mentioned. So I'm going to tell you about two projects today from SARS-CoV-2 and also our liver platform, which is based on a new protocol for creating liver organoids from induced pluripotent stem cells. All right. So this is what I was just sort of getting at is that both the advances on the experimental side in terms of the cellular systems that we can use with the microscopy, specifically high content microscopes. So one of the big advancements is in large 5 or 6 laser systems that have multiple cameras. So these instruments have like 4 cameras and they can do simultaneous acquisition. They're confocal, they have excellent Z resolution because they have immersion objectives. And so it's really amazing when you think about it. Basically it's a fully automated robotically controlled confocal fluorescent microscope. And so you can load anything from slides all the way up to 1536 well plates, and it will go to each spot, it will autofocus, and it will simultaneously collect a whole bunch of different color images, and then can do rounds and rounds of that. Of course, also can capture 3-dimensionality as well. So it's the confluence of this technology with machine vision, where we can do semantic and instant segmentation and do a really, really good job at feature extraction. So feature extraction is just simply the measurements that we're making at the individual cell level. And once we get to that stage, then we have human interpretable labels on our data. We can do all kinds of machine learning, deep learning approaches to, to understanding what we're seeing, because at the end of the day, we'll put one plate on our microscope and we'll end up with a terabyte of image data at the end of the day. And so, we're collecting millions of images, and we really need to have very efficient machine vision approaches. And luckily, the new advancements in GPU machine vision have dramatically sped all this up. So this has really only been possible for the last, I don't know, maybe 4 or 5 years, that this approach towards this kind of quantitative imaging and feature extraction. So it's really a time of convergence. So a little bit more about the experimental platform. So this is our Yokogawa CV8000. And so it's a big machine. It's about 3,000 pounds. And the plates go right here, and it actually, you can put compound plates here. So if you want to do live cell experimentation, you can do that. And so not only can you do live cell experiments and watch, with CO2 temperature and humidity control. So this thing also serves as an incubator. It can also do drug delivery. And so on the plate door on the right-hand side here is where you can put a compound plate. So you can do either fast or slow kinetics. There's internal robots for liquid delivery. So you can pick up a drug, you could start imaging, and then you can drop off a drug, and then you can keep imaging and tabulate a single cell result versus time, right? So, for pancreatic islets, for example, you can hit them with a calcium dye and then get fast kinetics and image with about 50 millisecond time scale. And then you can see calcium channels open and close. You can watch GPCR signaling versus time. So that's what we call fast kinetics. And then slow kinetics are where we can load a live cell plate and do a whole lap of imaging around the entire plate, which may take 30 or 40 minutes. And then we can come back with spatial registration and image that exact same area over time. And we've extended those studies out to 5 days. It's obviously limited by the proliferation. If you have cells that are proliferating, they'll just stack up and grow up, and then you can't see anything at the end of the experiment. But depending on how fast they are, you can do slow or fast kinetics. Justin, did you have a question? Oh, sorry, I thought you were talking at me. Yeah. So you can see data rolling in here. And so this is a 3-color assay, and this is just the speed with which 3-color images are coming in, and it's imaging 16 fields in a 384-well plate right here, right? So that's the microscopy platform. We got a whole bunch of lasers, and we can multiplex pretty deep, and we can even put 2 dyes on the same channel as long as they're spatially resolved. So if one's labeling the nucleus, the other one's labeling the cytoplasm or the membrane or something like that, we can do that and bootstrap ourselves up to easily up to 6 to 8 colors, right? And those can be anything from small molecule dyes to primary, secondary antibody staining to fish or RNA scope or something like that, right? All right. So the approach is to stain as many cellular features or compartments as possible to get as much information about the cellular system as we can. And then we use machine vision to identify each individual cell and or tissue or cells in tissue and extract features of interest. And then so that sets us up for doing perturbation experiments. And we're really a drug discovery lab. And so we have, we've been doing drug discovery for a long time and do de novo drug discovery, but we've been really focused on, since being at Michigan, is working very close to the clinic and developing, patient-derived disease models where we can characterize a disease in a healthy state and then screen compounds that are either FDA approved or have been through the IND process. So those are like clinical candidates. And we've got about 7,000 of those compounds on site now. So these are like really complex, very physiologically relevant, disease relevant, disease models, and then coupled with highly annotated compounds. So at the end, what we get is hits that have potential for translation to the clinic, which we've done, already several times since starting in like 2019, as well as just gives you a sense for the molecular targets and pathways that are influencing your disease state. So kind of like a chemical genomics approach, right? Okay, compatible with cells, organoids, and tissues. And, you can do anything from, arrayed CRISPR to things like antisense oligonucleotides, of course, to drugs. And the downstream analysis platform that we've developed is very similar to single-cell RNA-seq, how you analyze single-cell RNA-seq data, where we get a cell by feature matrix, and except that our data is not sparse; it has no sparseness. So we don't have, so our data matrices are very, very complete. Okay, just a little visual example of a straightforward image analysis, segmentation, and feature extraction. So this was from a screen that we did not too long ago. This one was a liver-focused screen for hepatic steatosis, and we've got an image of just the nuclei and the cytoskeleton here. Now, these are just our fiduciary labels for those regions of interest, for the nuclear compartment and the whole cell. And when you subtract those two, you get the cytoplasmic compartment, right? And so the image, the nuclear identification usually happens first because they're always fairly well separated. And then you can dilate those out to the edge of your secondary counterstain for the cell mask. And now you have two regions of interest for every cell that you can go about and and make measurements. And so there are biomarkers. We're multiplexed in this assay, so we're looking at brightness. We're looking at the radial distribution or the spatial distribution of the signal inside the cell, the texture of the signal. Is it smooth? Is it punctate? All kinds of different things that we can extract. And so this was a 17-plate screen, was about 5,000 drugs and 9 fields per well, 4 colors, about 1.7 terabytes of data. And the output data was 2,000 columns by 8 million rows. So the 2,000 columns are those are the features, right? And the 8 million rows were the individual cell observations. There are, of course, lots of highly correlated features, and about 500 of those survive after feature pruning for low variance or for high correlation, right? And it resulted in a CSV that was about 150 gigabytes. So the text data is about 10% of the total image data, which is pretty amazing. I always thought that the text data, normally the text, you walk up with a cell plate and you drop it on a plate reader and you reduce all that biological complexity to like a few kilobytes, right? But with this approach, it is a reduction, but it's not huge. So anyway, after we have our cell by feature matrix, we do the standard preparation for machine learning, where of course we do plate-based normalization. So in a 384-row plate, there's lots of real estate. So like columns 1 and 2 and 23 and 24 are usually reserved for controls, and those can be either different biological conditions or treatment versus no treatment or infection versus no infection, something like that. And that grounds us and gives us a tool to do batch to batch, correction, right? And then we center and scale features, by subtracting out the means and setting the variance to z-score to 1 for all the features. We also do a bunch of feature transformations. A lot of the intensity features are log normal, and a lot of the area shape measurements need to be square root transformed. And so there we've got good distribution qualities, and then that's when we start doing, model fitting, where we, do standard cross-validation and inspect model performance and everything like that. So I'll show you a little bit about this now. This was a recent example from my collaborator, Joshi Alamkal, who is in the cancer center. He came to us. This is the great thing about being at Michigan. This is why I love Michigan. First of all, I've made a whole bunch of friends here. But second of all, people come up to me with these incredible observational models, right? And basically all they need is they need a little bit of help to get it into a high throughput format. And we can then use this technology to look at a particular look for a particular disease phenotype. So I won't go much into the biological detail, but this was a nice illustrative example where we started off with an observation that the cell phenotype was in a particular state, and we wanted to flip it to a different state. And these were like known states, right? It's kind of a lineage plasticity sort of a framework, and but didn't have any way to do that necessarily. And so what we did was we did a small pilot screen of about 1,000 compounds, right? And so if we don't have any controls, this is the normal way that we approach this. We just throw some chemical probes at it and see if we can coax out a phenotype. And so this was just a two-dimensional plot of a couple of the biomarkers that we were interested in, and they were supposed to go in a particular direction. And this was hypothesized, right? And so what you can see is that biomarker 1 on the y-axis and 2 on the x-axis are doing something different. So we saw a whole pile of inactive compounds. We also saw a pile of cytotoxic compounds that were making all the biomarkers go down. That's one of the key aspects of doing this kind of screening, is that it's really nice when you have biomarkers that are going different directions, because it's really easy to get confused when you're doing drug treatments with general cytotoxicity, where if you're looking for the diminution of a signal, then sort of like, when this, this is what we call the cyclohexamide effect, right? If you just shut down transcription or translation, then that you can get confused as a false positive. So we have to have a really keen eye for what cytotoxic looks like in every single assay. And here the bubble size is mapped to the cell count. So we get an implicit, in all of these experiments, we get an implicit view of the cytotoxicity of the treatment, because of the fact that when the cells are undergoing cytotoxicity, they die, they detach, they get washed away during the fixing and staining process. And it's very well established that it is the most robust indicator of drug-induced cytotoxicity, is that the cell count just goes down. So that is something like reduction in viability. So we're definitely not looking for that. That's where all the signals were going down. But what we did find is we found a couple of outliers here, and these were two related compounds, so causing the cells to really change phenotype significantly. Now, this is just a bit of exploratory data analysis. So what we did is we ingested all of the cell level data and did a normal Euclidean distance calculation. We also did UMAP embeddings to look for discrete populations and everything like that. But this approach really worked out nicely. So this is how we found a good positive control that we could train a model on. So just going forward a little bit with the machine learning part, since we found this compound that was a strong candidate for a positive control for a larger drug screen, we wanted to investigate how what is happening, how the cells are being perturbed, right? And the other thing that we're interested in is we're interested in using this for discovery. So that means it needs to be statistically robust. So if you take the leading feature, the feature that was changing the most between these two groups of untreated versus drug treated, you can see that there is the negative controls here. So this is just the vehicle treated control, are the red dots down here. And then the inactive compounds you can see are the blue dots. So the blue dots are any compound treated well, right? And then the positive controls up here. And you can see that while there is a very nice separation in the signal, it's still a little bit noisy, right? And the noise is really confined to the positive control. So what we do is we run a random forest model. So we've been using the H2O.AI's AutoML. So this is a really neat way to survey a whole bunch of different machine learning model types. And it will run dozens of model types and will make you a leaderboard that will show you the model performance. And this is a super, super easy and easy to use Python package that will get you on a model very quickly and easily. So this particular model is an XG boost, and then what we want to do is we want to go look at the feature importance, and that tells us what is changing in this, what this model is picking up on, right? But feature importance, or the variable important features, VIPs in machine learning, in these kind of forest-based learners are they're not that easy to interpret because a lot of these features are redundant, and so their individual importance is being diluted by another measurement of that same biomarker. So this is not really a good presentation of the actual feature importance. What does work is this SHAP analysis. So this is shapely additive explanations. And this is, if you have two clusters, like if you're doing a single cell RNA-seq, or if we have, known conditions here, this Shapley analysis is it's just amazing to illuminate what are the true differences between the groups. And what this does is it uses a game theoretic principle where it scores models by leaving features out and only including other features, and it does this in an automated way to then actually tell you what the important features are. And so we saw that the one feature that was most important was bubbling up at the top here. That's the one we chose to investigate a little bit further. But when we actually fit the model, you can see the impact on the positive control by folding in more features here, right? So this model was folding in, a whole bunch of features, but really the only features that had significant weights were these like top 6 or 7 features that you're seeing here. And the main thing that this machine learning did for us was to enhance the robustness and the sensitivity with which we can detect compound efficacy, right? And really, that's the whole name of the game here. We're testing one compound at one concentration, and we need to have the best predictive power. We need to be able to identify the true positives. Now, false positives aren't a big problem for us because here we're picking compounds that we do, dose response follow-up, right? So we'll do concentration response curves, and any false positives will generally drop out of that step, which is why we call it the confirmation step, right? Okay, so that's a little bit about how we use a random forest model to basically score an assay. And so we have an assay metric we call Z prime in high throughput screening, and this tells us what the difference is between positive and negative controls. And if you're doing really good in a cell-based assay that has high biologic heterogeneity, if you get to a Z prime of 0.5, it's really amazing. And 1 is perfect, where you have a perfect separation between your controls and no overlap. And this was like a 0.95 Z prime or something like that, just ridiculously high. So we can, of course, go through and make some violin plots for the interesting features. And then this helps us interpret what the model is calling, so it's not a black box model, right? Then the last thing here is that once we have a score like this, we can go and see how it manifests. So here in this linked dot plot, I've just got a couple of plates worth of data here. And this is our machine learning score. If you lasso that data, you can see that all of these are producing a very high distance score. And then you can see how they manifest in terms of cytotoxicity. So that there's a lot of these high scoring machine learning on the machine learning model are also producing a large phenotypic distance, meaning that if you look at the image, they look different. And a considerable amount of that is due to cytotoxicity, but not all of it, right? And so maybe what we're most interested in is when we're getting these phenotypic perturbations, but it's not linked directly to just the cells dying, right? So that's this is a framework that we use to help score cell-based assays when we're screening. Okay, so anyway, thanks to Joshi Alamkal and his group, for this wonderful collaboration. And so this is a work in progress, and so I'm going to tell you about another application of this in SARS-CoV-2. So in early 2020, after we had assembled our drug collection for our drug repurposing center, we set about to use this technology to do a drug repurposing screen against SARS-CoV-2. So this involved recommissioning our biosafety level 3 facility, which we have. It was functional. It was just nobody had active protocols, and so we had to recommission, inspect the facility, get it up and running, validate that everything was functional, and we fairly quickly were able to obtain live virus from BEI resource and get, get viral propagation happening and get everything that we needed to conduct this screen, which was based on the authentic infection in a human cell system of SARS-CoV-2. And there were a couple of groups working on similar approaches in parallel, but we're using, non-human primate cells, and we're using simpler endpoints like protection of a cytopathic effect. And so we chose a different approach that would allow us to, gain some insight into the mechanism of action. So this is the this is the timeline of the final assay. We plate cells in our BSL-2 lab. We screened dozens of cell lines, but it turns out that HUH7 gave the most robust, reliable result. It's a liver cell line, and it's known for, for sustaining like hep C infection, and it's been used in context of a few other viral infections. So it's permissive generally to certain viruses. And so that's what we chose for screening. And we would dispense drugs, plate cells this is done in the BSL-2 drug discovery laboratory, right? Then the plates go into the biosafety level 3 lab. So, full respirator suits, PAPR respirators, Tyvek suits, and high containment. And then they go in, they get infected with SARS-CoV-2, and then they incubate for 2 days in, just in a regular CO2 incubator in BSL-3. Then the plates come out, and, no live virus can leave the facility. So after that, they have to get fixed and then and surface decontaminated. So the fixation fixes anything in the well. And this is just with paraformaldehyde. So this is 4% PFA. And then the rest of the plate gets surface decontaminated. We log the samples out. They go into secondary containment. They go into the biosafety level 2 lab, and then there we do the staining protocol. So this staining protocol, we had I'm just showing you a couple of channels here, but we had we stained the nuclei with DAPI. We stained the nucleocapsid protein, the SARS N protein. We also had antibodies working for the S protein, for viral RNA, and a couple of other endpoints, and also lipid droplets. So there's a well-known lipid droplet effect here. So we stained neutral lipid droplets as well. And so we had those endpoints, and then we're able to do a simple pass of scoring for efficacy, where we're just simply using the machine vision to identify infected patches, and then figuring out what percentage of those cells are infected, and then looking for a statistically significant difference there. And then we'll pick hits and then confirm in dose response and try to gain some insight into the mechanism of action. And so what was really stunning to me is that is the amount of phenotypic diversity in the well here. And so what you're looking at here is a false colored image of just the nucleocapsid protein. And so I see all kinds of different cell phenotypes here. I see small round with a negative stain in the nucleus. I see small with fairly homogeneous staining across the whole cell. And then I see the guys like this who have, these noodly appendages, right? So these are tunneling nanotubes and Philopodia, right? And so this is a whole field of cells. And so what we're observing here is we're observing the progression of the viral life cycle inside the cell, right? Inside this cell culture population, because there's a couple of generations have happened here. And so the point was not to just turn this into a static number of like how many cells are infected here, but to capture some of that phenotypic diversity here so that we could score that and then look for perturbations that might be clinically relevant also. Like, for example, this is what's called a syncytia. A syncytia is where you get one cell infected, and then it propagates to a few other cells in the neighborhood. And then what happens is that the whole plasma membrane gets remodeled to form one giant multinucleated cellular body, right? And this is directly observable in like CT scans in patient lungs. And so this happens in lung tissue. And so we hypothesized that, look, we could interfere with this viral life cycle at everywhere from before entry all the way to replication to this formation of syncytia where other assay technologies without spatial resolution would just not be able to see any of that stuff, right? Okay, so, and this is an example of some dose response curves. So these are curves where, well, if you look at remdesivir here, so the red curve is the efficacy curve, and the black curve is kind of what I was talking about before with cytotoxicity. So what you can see is that as the concentration of the drug is increasing, this is a good signature here for entecavir, remdesivir. We see a bunch of daylight between the efficacy curve and the viability curve. So you can see the viability is staying high. So this gives us an implicit cell-based therapeutic index, right? We don't want to pick compounds where both of these curves dive at the same time, because that's just the cytotoxic effect. And a lot of groups published those where they got confused of that. And that's what we call the bleach effect, right? If you kill the host, viral replication shuts down really, really quickly, right? So, okay. So this was a fun experiment that gets to some of the spatial analysis where it was a time series. So we did this early on to gain information about the progression of the phenotypes through time, right? And so in a 384-well plate, we have uninfected, and then we've got, you know, where, so this was a nuance that was important. We plated the cells all at the same time, and then we took it into biosafety level 3, and then infected, staggered in time. And the other way to do that is to do a whole bunch of plates and plate them all at the same time, and then, I mean, is to do it the other way around, where you have separate plates where you're stopping the viral infection at a different time. So you plate the cells all at the same time, and then you fix, the couple of columns at a time. But what we realized is that when you do that, the total amount of time that the cells are in the dish is different. And the ones you fix very early are sparse, and the ones you fix at the very end are very dense, leading to a bunch of confounding phenotypic differences, right? And so this is a better approach, where instead of doing that, what we did is we plated all the cells at the same time, and then came in and staggered the infection. So the clock time for how long the cells had been in the plate was the same at the very end of the assay. The only thing that was different was the amount of time they were infected. And so we, of course, did imaging, feature extraction, data processing, and model fitting here. And this was another random forest model. And we used the time point as the ground truth here. And what you can see is that we've got incredible predictive accuracy. When we can look at an individual cell, we can tell how long it's been infected for. And we can use this as a model of pseudotime, so that when we treat with drug, we can go specifically look for drugs that are arresting the viral pseudotime at a particular time point. So early would be entry, in the middle would be replication, and at the end would be some of these weird phenotypic, alterations that SARS-CoV-2 does to, human cells. And so what starts to emerge here is that we see a progression, and this is what the progression looks like. So you start off with these early infected cells here, where it's punctate, staining pattern, and dim, right? And that makes sense. So the virus gets into the cell at time 0 there. Once it gains entry, there's really no expression of viral protein. So that takes some time. Then we see a brighter punctate staining pattern in isolated cells. Then what we see is we start to see the cells forming protrusions. And again, this is, a cell-to-cell infection mechanism that we're observing here that leads to a bunch of cells that are adjacent but infected. And then given more time, that's where we form these really creepy syncytia, right? Where you get these big monster, multi-nucleated cell bodies. And when we treat with remdesivir, you can see what happens. There's a very characteristic spot, or a couple of spots. So this is arrested, at early infection. And so it just, remdesivir inhibits the RNA-dependent RNA polymerase. And so, the virus gains entry, sets up a replication site, but really doesn't proceed from there. So that's an example of how we can use this to find something that's interfering very early on in viral infection. So what else I wanted to say? Yeah, that's about it. Okay. So if we take this cell population and do the UMAP embedding. So this was, thousands of features measured, but after feature pruning, this was like 600 or 700 features. All of the cells presented a UMAP embedding that looks like this. So it's a distinct and distant cluster, right? This is got to be just the main cell body here. And there's a couple of other little clusters, but we didn't really know too much about this until we started poking around a little bit. And so a great thing to do here, and this is really fantastic because unlike you people who do transcriptomics, every one of our data points here, you can just pull up the image and look at the cell and like see what it looks like. And so that's a really powerful thing for us, in that we can just lasso a group of data points, and then it will pop us the individual cellular image of what those cells look like. So when we treated with controls, you can see here that we've got the clusters color coded. When you treat with remdesivir, this is the infected cell cluster. It's disappearing, right? And but what we were interested in was how much structure is there in this infected cell cluster. So what we did is we extracted this cluster and then re-embedded it, right, to spread it out a little bit. And this is what we see. So just as I was showing you before with that viral pseudotime model, all of these distinct clusters emerged. So this is a fantastic method for identifying phenotypically distinct morphologies that exist inside a cell culture population, right? So we've got syncytia. So we had this distribution from single cells all the way to, now these aren't in order or anything in time series like they were in the ML model, but what you can see is that there are small dim cells, bright dim cells. There are cells with a bunch of spaghetti hanging off of them, and adjacent infected, and then big syncytia, just like we saw before. And what's really cool is that there are all the spatial relationships are preserved, right? So you can see this is in the center here. This is a cell that belongs to a mature infected cell cluster, and it is surrounded by early infected cells, right? And so that's just kind of intuitively obvious here. When you look at it in retrospect here, you can see that this is a big cell, and he's nice and bright, and he's surrounded by cells that are have this dim punctate staining, right? And there's one other guy here that has a little bit more. So, this guy reached out, infected that guy, and then these guys were even another generation of infected cells. So pretty cool. So this is really what emerges. This is a messy slide, but this is this global UMAP embedding of isolated infected cell morphotypes and multicellular bodies, and yeah, so that's the characterization of SARS-CoV-2 infected cells. And so we use this to do the drug screen, and then just show you a couple of dose response curves here. There were we reported, I think, 17 drugs here that passed our test for potency for drugs we felt that were not a spurious effect, and or drugs that were just interesting and other people were reporting on. And a couple of them are that I want to point out is clophazamine. It's an interesting old antibiotic that's been used for leprosy, also known as Hansen's disease. Metoclopramide is a GI drug. Lactoferrin is a commonly available dietary supplement. And we actually have 2 clinical studies ongoing now for lactoferrin. And ipratropium bromide, which was incredibly potent. And also this is could be kind of like a 2-for-1 scenario where this is also, the brand name is Atrovent. So it is a bronchodilator, and it's delivered directly to the lungs through a rescue inhaler, right? Or a nebulizer. And so we're in the middle of doing the electronic health record mining to look for patients who are on Atrovent and compare them to a comparable cohort of patients who happen to be on albuterol. So albuterol was also in our experiment, but albuterol did not have any antiviral efficacy. So that way we can at least normalize out the patients with COPD if they have a different, or asthma. They may have a different disease progression because of their underlying comorbidity. So anyway, that's a work in progress. And then just one more before I shift gears and tell you real quick about our liver platform. This was fascinating. So this was a collaboration with Arul Chanayan. We'd been working together since the beginning of the pandemic. When we established our SARS-CoV-2 infection models, they've known for a very long time that that androgen signaling affects both ACE2 and TMPRSS2, which are required for infection. And if you treat with some prostate cancer drugs, that should cause ACE2 and TMPRSS2 to decrease, and that should confer some efficacy. Well, indeed, that happened, and we saw a resolution, an almost complete resolution of the infection for most of these drugs. The best ones were enzalutamide and proxalutamide. And enzalutamide is FDA approved for prostate cancer, and it blocks the androgen receptor. Proxalutamide is a next generation, same sort of drug, but it also degrades the receptor as opposed to just inhibiting the receptor. And this one is proxalutamide's in clinical trials, phase 3 trials for prostate cancer right now. It's being developed by a company called Kintor. And they went ahead, they saw this data. They went ahead and they initiated a couple of COVID clinical trials. And at this time, there wasn't a whole lot of infection in the United States. It was really raging in Brazil. So they did these clinical studies in Brazil. This is in an outpatient study. So this was 134 patients in the proxalutamide group, and in the control group was 128. And they looked at the hospitalization. This is split by males and females. And you can see that the hospitalization rate went from 27% to 0. And then in females, it was lower to begin with, as we know. Then the hospitalization also, very dramatically reduced. But what was really stunning was they administered proxalutamide to hospitalized patients. So these patients are being admitted. Generally in Brazil, they're being admitted with more disease severity. So on the COVID-19 ordinal scale, they're here we're probably admit on a 4 or a 5. They're admitting under worse, at higher scores there. So these patients have severe disease. And what was really amazing is that, and this is fairly significant numbers. This is almost 300 per arm here. They saw that the mortality at day 14 went from 47% to 3.7%. And the hospital length stay went way down, and the need for ventilation went way down. Now, this study has been under scrutiny, and it dramatically needs to be repeated. But it's a really interesting potential therapeutic modality that should be invariant to what strain, and because it's modulating a host factor. Anyway, so that's just some exciting result from this clinical trial based on proxalutamide. So, yeah, okay. I guess I'll just move on to the liver slides, but this is just the summary here is that we can use this kind of morphologic cell profiling to identify compounds that have efficacy, and we can characterize their effect on the disease model in terms of what kind of perturbation a particular drug is doing. And we can fairly quickly understand whether that's a positive or a negative thing, and then go and focus on what we think is positive. And so this is why this style of approach has a much higher translation to in vivo efficacy over conventional drug discovery techniques. Cool. All right. Okay, so I want to tell you about one more project here where we used this kind of technology to model human liver. And so we developed a multi-omics liver platform using a protocol that was published by Takebi in Cincinnati at the end of 2019. And so what this entails is forming human liver organoids. So starting from induced pluripotent stem cells, growing up these organoids. And then we developed a couple of different things. We developed a high throughput screening platform so we could screen lots of compounds. We also developed a organ on a chip system for more physiologically similar environment. And the endpoints are biochemical assays of, we're initially using this platform to investigate drug-induced liver injury, which is a big problem in drug development because the majority of clinical failures, either in phase 3 or in post-launch for recall and black box warning, are due to unanticipated or idiosyncratic drug-induced liver injury. And so, it's a huge societal cost. So, and it's also was a fairly straightforward platform to develop, as opposed to a more distinct disease model like fatty liver disease or fibrosis, which we're also very interested in. So we've got biochemical assays, we've got our entire imaging approach, and then we've incorporated single cell RNA-seq. And so these organoids start off as iPSCs, so we can get from a blood draw, we can isolate CD34 positive cells, reprogram them into iPSCs, and using Jason Spence's protocol, going from iPSCs to foregut spheroids. Then this is where the TKB protocol takes over, where it's really just treatment with retinoic acid and hepatocyte growth factor. Then we can get to this human liver organoid stage where we form from the same individual hepatocyte stellate cells and the liver resident macrophages called Kupffer cells. And this is really amazing because this has never been done before. The conventional models are either cancer cell lines. There's a couple of other things like HepaRGs, which are not cancer, but still proliferating cells. And then there's primary human hepatocytes, which are just terrible. They're so hard to experimentally control. You can only keep them really metabolically active in culture for 12 hours or something like that. And then you have this massive donor to donor variability, not to mention the circumstances under the donation. These are from generally from cadaveric livers that were either discarded from the transplant list or that's the main thing that happens is if they're if they were not eligible to be transplanted, then they get diverted to the research platform where they can be isolated and sent out to people like me, where we can plate them, and they require all this matrigel base and then a matrigel overlay, and then they still tank after something like 12 hours. So really just not a great experimental solution for that. So this has really limited the field of liver disease for a long time. And again, Charles, Sophia, and again, Matt, we're sort of the liver team here. We've been developing this work. And we've, of course, developed imaging approaches. So this is, we've done 3-dimensional imaging, looking at all kinds of different biomarkers. And so this is in the intact organoids. Then what we can do is we can take the organoids, disperse them, plate them, and then expose them to drug. And then we can do the same thing I was showing you before. We can look at biomarkers like NF-kappa B translocation, reactive oxygen species, lipids, mitochondrial membrane potential, ER Golgi status, and then just cell death. And then can take all those features, project them down. And now we can get clusters that are specific to, the drugs and the drug mechanism of action, which is really cool. So, so that's our 2D platform. And we have a library of drugs that cause drug-induced liver injury. And this particular individual responded to these drugs. And so the red curve here is HOH7, which is a very commonly used liver line. And it just doesn't respond to all these drugs. So it's just not a good model. And when you look at these cells, they're much more sensitive to drug-induced liver injury. And the reason why is because they have a true hepatocyte, metabolism. So, okay, now we took these organoid cells to get even a more, organotypic sort of platform. We use this system for growing organs on chips from Emulate Bio. They're a Boston company. They have this platform. It's just been commercially available not too long. and this is called the Zoe. And so it has a gas regulator and then this chip system where the chips snap in and you can add different medias. And so you've got inlets and outlets, and it has a upper chamber and a lower chamber that's supposed to represent, vascular, sort of arterial and venous flow. And you can control the medias in each compartment. But really what this does is this puts them under physiologic flow and then helps them become more differentiated and gets rid of the immaturity. So the evidence of that is the fact that they produce a lot of albumin. And so this is the organoid chips alone. So, regular cancer, liver cancer lines don't do that. The other thing that the liver cancer lines don't do is they don't produce drug metabolizing enzymes like the cytochrome P450s. So here we were just gobsmacked when we saw that, first, if you ignore the big blue bar here, if you just look at the purple bars versus the other cancer lines, these human liver organoids, as compared to HOH7 and PH5s, where most of the time these SIPs are undetectable, we were like high-fiving each other when we saw, wow, this is a tremendous difference. And then we put them on this chip. We put them on the chip, we just got these massive induction of cytochrome P450 expression. Really, really amazing. And when we treat them with DILI drugs like Tylenol and thioluridine, we see what we would expect. Now, these are the biochemical readouts. We also have these imaging-based readouts as well, because these chips come off and they go right on the confocal microscope. We can even do this in a live cell context because the chips can come out and they can go right back in to the emulate system. And so you can see there's a semi-permeable membrane. So there's a pattern of holes in the background here, and we're looking at the top chamber, the hepatocyte chamber, and you can see that this is what normal looks like. Acetaminophen is characterized by patchy necrosis, and thioluridine is characterized by hepatic steatosis, where these cells are accumulating lipid dramatically. And so I think we're probably should wrap up pretty soon here, and happy to entertain questions. We did point this at a drug called inirigavir, and we were successfully able to reproduce the clinically observed drug-induced liver injury. So this was caused a failure of a phase 2 clinical trial where 30% of the patients experienced drug-induced liver injury. One patient unfortunately died. None of the models predicted this. So what we did is we ran these experiments and successfully predicted that this combination was bad. So if the FDA would require this kind of experimentation platform as evidence for drug-induced liver injury, this drug may not have entered the clinic. And so this is, there's been a big debate about this, about how well animals recapitulate human drug-induced liver injury, and they really just don't do it very well. And this is one of those cases where there was small animal, large animal, and all the cell models, normal cell models for drug-induced liver injury, they just did not recapitulate that. So, okay, the final thought here is that we've done single-cell RNA-seq on on cells that have been cultured on this chip system. We lifted them up, treated them with drug, lifted them off, and did the single-cell RNA-seq. And so we're in the process now. We've already written this up, and it's up on bioRxiv. I'm showing the preliminary results of the single-cell RNA-seq that give us generates hypotheses for the mechanism of action for this combination of inirigavir and tenofovir. These are hepatitis B drugs for chronic hepatitis B. And we also have the corresponding phenotypic data. And so one of our big goals here is to align the single cell transcriptomics with the single cell phenotypes so that we can learn about the transcriptional programs that are different from that are going on in these morphologically distinct cell clusters, and then look at things like the spatial effects. And so we see this in our culture system. So when we're growing these cells, we see patches of hepatocytes. Now, this was Junhee Lee's paper on seq-scope in the liver, showing that they could see differences between hepatocytes in zone 1 versus zone 3. This is the big difference between the zones, is that oxygen tension is comes in at the artery, right? and it's low at the central vein. And so, as you would imagine, glycolysis happens in the oxygen poor environment, and beta oxidation happens in the oxygen rich environment. And so we have an initial sense from the gene lists from Junhee Lee from this paper that we're observing some zonation and some metabolic specialization inside this culture system. So really exciting stuff, and we're still working on this, and hopefully there'll be a follow-up paper on this. But with that, just like to say thanks for your attention, and happy to answer any questions you may have.
That was great. Thanks so much, Johnny.
Yeah,
so
thanks.
if folks have questions, feel free to type them into the Q&A. We did have one question in that I think is a good one. I also was thinking about this as you were talking. So Kathy asks, how do you automatically identify multinucleate cells? So normally each cell is defined by a single nucleus, but multinucleate giant cells don't follow that rule.
Yep. We've got several different strategies. It really depends on what biomarkers or what molecular probes are in the experiment. So for the SARS-CoV-2 assay, that was a big distinction. So normally what we do is we identify nuclei first, right? Now we've got a set of nuclei, and then we look at the secondary stain, at the counter stain. And that counter stain could be a cell mask stain, could be a cytoskeletal stain, also could be a plasma membrane stain. So this works really well with plasma membrane stains. So you have a nucleus, and then you dilate out, you extend that nuclear region until you hit the plasma membrane stain. And if there are multiple nuclei inside there, then you will be able to identify that, right? Another way to do it, this is something we had to do for the SARS-CoV-2 work because of the syncytia, is that the first thing that we did is we identified the virally infected patches, right? And then we identified the nuclei inside. So that gives us a relationship between infected patch and the nuclei contained within. And because of the way the staining works, when we stain for the N protein, if you have two distinct cells, like they still have a plasma membrane separation, there is a prominent valley of intensity in between the two cells, right? So in that way, we're able to identify those syncytia by first identifying all of the single cells based on the N protein staining, right? And if they have valleys in between, they get separated as individual cells. If there's no valley in between, it just becomes one large cell body. And then we count the nuclei inside. So a single infected cell has a N equals 1, and then a syncytia is anything greater than that, where the greater the number of nuclei, the higher confidence that we have that it's actually a true syncytia.
Cool, that's great. I guess I have a question about your liver organoid system. So we were just in our Society of Toxicology meeting last week where we were hearing about how the iPS-derived organoid models are great and you have patient-specific data, but you have this like immature phenotype, which you were referring to. And so now you've taken this to the next level with these organ-on-chip systems. I'm wondering how far are we from, modeling this intersection between a disease like NAFL and drug toxicity with these organ-on-chip systems?
Yeah, it's a great question. So, so if we're talking about DILI first, drug-induced liver injury happens between 1 in 1,000 and 1 in 10,000 people. And there's specific genetics that influence that. This comes out of the GWAS studies. And so just serving any old individual is probably not good enough, right? So we have an approved IRB, and we have funding from NIDDK to go about and establish a biobank from patients who have experienced drug-induced liver injury, right? So now we're loaded with those genetics, and we're this initial pass, we're going to be focused on the worst DILI drug, which is Augmentin. So Augmentin, if you have kids, you've probably given Augmentin to your children for ear infections and that sort of thing. So Augmentin is a drug combo between amoxicillin and clavulanic acid, and it causes DILI in maybe 1 in 1,000 people. So we're going after that patient population. We're going to go after. We're going to build 10 lines from 10 different patients, who have experienced DILI with a focus on Augmentin. So that's what we're doing for that now. Towards the other diseases, we have developed these into disease models for NAFLD, and we can do that one of two ways. We can take this cell population and we just make them fat, and we make them fat the same way that I got fat during the pandemic. It's just overnutrition. So we can feed them excess oleic acid, conjugated to BSA, and then they ingest that, they become fat. Or we can stimulate de novo by hitting them with high glucose, high insulin, and an LXR agonist. And then they become fat. And so they become turgid with lipid droplets. And we can count the lipid droplets and look at the total fat content inside the cells. And that's a great model for hepatic steatosis. Now, let me see if I can go back to this image here. For the progression, so what happens next after NAFLD is fibrosis. And this model, take it or leave it, it forms a non-physiologic complement of cells. So we get about 60% hepatocytes, but 30% of the cells that we observe are hepatic stellate cells. So these are myofibroblasts. So that's not great. It starts off out of the gate as a model for NASH, for non-alcoholic steatohepatitis, because we have activated stellate cells here that are, positive for alpha SMA, and they're making collagen. And so they look active, and the way to get rid of them is to sort them out. But their presence, can be used directly as a disease model for stellate, stellate activation. So yeah, we also have our eye on that. We're going to develop that into a more robust model. But the stellate cell, presence of the stellate cells haven't been confounding our endpoints for the hepatocyte biology so far, but it is on our bucket list that we do need to do some magnetic bead sorting to sort them out so that we can get a pure hepatocyte population. We'll obviously have higher signal to noise because we'll have more hepatocytes present that are doing more of their job. So that's how we went after DILI first, but then are extending those to other, the diseases that we're most interested in, which are non-alcoholic fatty liver disease, and that transition to forming scar tissue, which is NASH.
That's great. Yeah, sounds like some really good future directions. So yeah, thanks so much again to Dr. Sexton for this fantastic talk. Really appreciate it. And we will see everybody at our next seminar. Thank you all for attending. Have a nice afternoon.
Thanks, everybody.